Okay, good morning, everyone. I think we can start. So I just need you to get back to your microphones, switch the cameras
Morning.
off, if you can. So the first question is, can you hear me?
Yes, we hear you.
You can hear me, but I cannot hear you. That's the problem. Okay. Okay, now it should be better. Okay, I'm sorry. I forgot about this one small thing. Okay. Today's presentation and the last part of theoretical information and the background behind our laboratories. Maybe we can start from your labs. So, was there anything that you did not understand fully, or you have questions, or you disagree with something during the procedures? So was your okay? Yeah, the question is whether you've seen your result and you understand the result of Western blotting.
Yes, we have seen it.
You saw it, okay, because for sure Group 6 will discuss the results during the next classes, because I saw it after the laboratories. So I just want you to pay attention that there are many bands of the protein, which seem to be specific when you compare it with the negative control. And this is normal in case of this protein, because when we have membrane proteins like CD81, it may happen that they form different forms, migrating at a similar but still a different size. So depending on the conformation of this membrane protein, you may expect it as migrating in different bands. Plus, you may still have free GST. So please compare anti-GST and anti-CD81 blots. If, for example, you can see a band, well, it should be quite strong, which is GST positive, but CD81 negative. This should be free GST. We don't fully understand why it happens that still free protein tags are produced, like free GST in this case. Apparently, there is some kind of termination of translation, and only GST is produced. Or maybe there are still some proteases in bacterial cells which recognize a similar sequence to the one that is between GST and your CD81, that it releases this small protein. But this is how it goes. I mean, when we have fusion proteins, it is very, very often that we have free tag protein still present, plus the fusion protein, of course. Okay, so this is it. I hope that you could notice that the solubility of your protein is not great. So we have still some in the soluble fraction, but dominant, I think dominant amounts are in the insoluble fraction. So apparently they form inclusion bodies. It all depends on the culturing conditions. I mean, maybe not all, but many depends on the culturing conditions. So this induction was quite long, 5 hours at 37 degrees. Maybe this was not optimal. Maybe when we do it at room temperature or 30 degrees overnight, it will give better result, okay? So this is it. Are there any questions that you have? Now everything was clear? Okay, so I have a very basic presentation for today. This is just to remember all the things that we should say about. Okay. Okay, so apparently my computer doesn't like my presentation. I will try again. One second. Okay. Can you see the presentation? Or it's
Not really, only black screen.
Not really black screen. Okay, so I have to do it different way. I have to upload it to Teams. Okay, so this one is open. Just my computer doesn't see it. I don't know why. Okay, can you see the presentation now?
We see it now.
Okay, great. Nice. Okay, so we are supposed to talk a little bit about protein tags, or just refresh our knowledge on protein tags. So the first question is when we will use affinity tags, like here we have GST. So what's the reason of adding tags to our target proteins?
To make isolation of overproduced protein easier?
Okay, so it's for purification or pull down experiments. So we want to get to our, so we want to purify our protein or to get interacting proteins using this system. Another reason.
Maybe in some studies like the hybrid system in yeast?
Okay, but this is a very specific situation. So again, interactions. It's quite true for GST usually. Well, we don't see it too much, maybe.
To increase the solubility?
Yes, to increase the solubility of a protein, especially bigger tags are used like that. So we choose proteins which usually fold quite well by themselves, or they don't need too much help from the chaperone system. So then they are used as tags. Another, I can see at least two more reasons. So think about it, and not only one person. So let's imagine a situation we don't have anti-CD81 antibodies. Generally, it's a new viral protein, new virus. We don't have any kind of antibodies against the protein. So then we target what for? So if we don't have a specific proteins and we still want to detect the protein, then we tag for detection, okay? Because protein tags usually have nice commercially available antibodies that can recognize the tag itself. We can use it for detection in Western blotting, but also to analyze localization of our protein. For this reason, we may use fluorescent protein tags like GFP and related proteins. So this is for detection, localization, transport, analysis. Plus, there is one more reason as regards usually small proteins. So if we want to work with a small protein and we want to use it for immunization, small proteins below 10 kD sometimes have very low antigenic properties. So they do not induce antigenic response, immune response very well. And if we bind it to a bigger protein, this forms the antigen, the fusion antigen. Okay, so we have a hapten, so not a fully capable antigen on its own, but when we link it to a bigger protein, it gets the antigenic properties. Okay, so this is the reason, for example, why if we want to immunize with peptides, I told it to my group. I don't know if all of you know how rabbit monoclonal antibodies are produced. Sometimes we can buy rabbit monoclonal antibodies, and it's a bit puzzling, like how you can get rabbit monoclonal antibodies. It's not by fusion of rabbit B cells, but these are anti-peptide antibodies. So they generate only one clone of B cells because the epitope itself is used for immunization in the form of a peptide. But the peptide itself usually is not sufficient to induce the immune response. So it is either chemically, well, usually actually chemically bound to a carrier protein, or it can be used as a fusion, for example, with GST. GST has very nice antigenic properties. It's also good for chemical linking because it has lots of lysine residues, and lysines can be used for chemical linking, for example, with polyethylene glycol, so PEG. And then GST can be also used as a carrier. So immunization, increasing antigenic properties. This is, let's say, the last main reason why we tag proteins. There are also some specific applications, like, for example, we may study ubiquitination of proteins. For studies of interaction using also proteomics and mass spectrometry, there are specific domains which can enrich the analyzed material with our protein. Okay, but we have GST and the schematic representation of what's going on our beads. So this week, what will be happening is here in this picture. So we have CD81, which is our target protein, fused to GST, and you have a lysate. You know that there is some small amount of the protein in the soluble fraction, so in your lysate, and you will incubate it with glutathione affinity matrix, which is agarose. So this is GSH, glutathione agarose, which is used as beads. Reduced glutathione is a substrate for GST, so it will be bound to the enzyme, to the substrate, the substrate with the enzyme, and we will fish out our protein of interest, and of course interacting proteins, if there are any, unspecifically or specifically interacting. We will use so-called batch purification method. So we are adding the beads directly to the lysate. There is no column. Of course, we can use a column, but with GST, batch purification works very well. There is no reason to use a column because we are not eluting with any kind of a gradient. Columns are good when you're eluting with a gradient of the eluent. So eluting a reagent, like, for example, when we have histag and imidazole, we may use an increasing gradient of imidazole in our buffer for elution, like increasing salt concentration, for example. Then we can use columns. But here we have batch purification. So the beads must be also washed. For washing, you will use a low amount of the triton containing buffer as a detergent to remove all the weakly binding, unspecifically binding contaminating proteins. And then we will elute our protein, so just separate it from the beads using a buffer with reduced glutathione. Okay, so this will be the elution buffer. And in the mixture then, in the eluent, we have our target GST fusion protein, but also glutathione itself. And we should remember that because depending on the application, so what we want to do with the protein, we have to remove glutathione or not. We will not do it during the laboratory classes, but for many applications, glutathione also must be removed from the mixture. Okay, this glutathione agarose also gives the possibility to remove the tag when we are using protease recognition sequences. So these are PGX series plasmids, and what you can see is that in most of them, between PGX, the GST protein, which should be on the left, and your protein of interest, which should be on the right, so there is N-terminally used GST, and our protein is at the C-terminus of the fusion. There are recognition sequences for proteases, like thrombin, like factor XA, so this is the clotting factor from our blood. And we have more specific proteases like precision, precision, I think, is a modified TEF protein, TEF protease. So it's a viral, picornaviral protease that is used. So they are like short, specific amino acid sequences which are recognized by the proteases. And we linked it to our protein, not by, of course, chemical linking, so not by protein to protein, but at the step of DNA. So we create genetic fusions. Then we have another question, how to create a fusion. So what should be remembered when we want to create a fusion? Well, of course, nowadays, I think the most popular way is just to order a synthetic gene. When you design the sequence and you just order it, you say the company what's the DNA sequence, and they will produce the plasmid. So they can clone it into PGEX and they will just send you back your fusion gene for expression. Then you can do bacterial transformation, like we will also do this week. But sometimes we just have to isolate our gene, so encoding our protein, and we have to fuse it, clone it to the PGEX series plasmids. So what we should remember, that glutathione has no stop codon, GST gene has no stop codon. So the protease recognizing the protease recognition encoding sequences are in frame with GST, and there are usually several restriction enzyme cloning sites where you can clone your gene in different open reading frames. So you can choose the one that will preserve the open reading frame, and your protein of interest will be in the same open reading frame with GST. Your gene of interest should have no start codon. Usually we remove the start codon. Although this is the bacterial translation, and it should recognize the Shine-Dalgarno sequence, which is in front of GST, but still it's the safest just to remove your start codon, so only ATG, and then you take the sequence of, for example, CD81. This is a fragment of the protein, so for sure without the start codon, and you add the stop codon either at the end of your gene, but in the PGEX plasmids there are also stop codons, usually in different open reading frames also in the plasmid, just to make sure that there is termination of translation, of transcription, and then the transcript is proper. Sometimes in between GST sequence and your protein, your target protein, there are also different kinds of linkers, apart from those protease encoding, protease recognition site encoding sequences. Because it may happen when we have fusion proteins, like tag proteins, that the tag itself interferes with the folding of your protein. So in order to ensure proper folding, we are separating the tag and our protein of interest by the means of a linker. And there are two types of linkers which can be used. The first type is the so-called flexible linker, by using amino acid sequences which are wobbly. Usually this is pentaglycine linker. There is also a PGEX vector which is called PGEX KG. KG stands for kinky glycine, so there are like 5 glycines, one after each other, and they form a flexible linker between GST and your protein of interest. So this can help with the protein folding and solubility. But sometimes flexible linkers are not good for a protein to preserve its function, and we use so-called rigid linkers. So linkers which are more solid, they have structure, usually alpha helical structure, and there are sequences which are known, which you can use like repeats of some motifs, which will form an alpha helix, a short alpha helix in between your protein and your tag protein. Okay, so if we clone our CD81 in one of those PGEX vectors, we have 2T, which is here at the top. So we have thrombin cleavage. Yeah.
Because I see still the first slide. I don't know if anyone has the same
problem.
Okay,
so they don't change.
Yeah.
No, they don't.
They don't. Okay. So once again. So instead of increasing my knowledge on Teams, it seems I have more and more problems with Teams. I don't know why. Yeah. Okay. Because I have it. Now?
Now it works.
Okay. So I have no idea why. Sometimes it works, sometimes it doesn't. Okay. So I was talking very theoretically. So now you can see the sequences. So there's really a lot of PGEX plasmids that you can buy and use. So we have thrombin, we have factor XA, we have precision. I think precision and TEF protease are now more and more popular. So usually I think people use those PGEX 6P versions of the plasmids, but we have an old PGEX 2T and we have thrombin recognition sequence between GST and CD81. So when now getting back, so how to remove now GST from CD81? For example, if you want to work only with CD81, we can, when we have the eluted protein, we can elute it, or sometimes even we do it on the beads. This is also possible. So we just add a buffer which contains thrombin or another protease for other plasmids, and it will remove CD81 from the column, and GST will still remain bound to the glutathione agarose. Okay, so this is how we can remove it, but then we have to remove the protease because it will be in the solution. So proteases are sometimes also tagged, and you can like fish out the protease then from the solution. So that's how it goes. Okay, so as regards the fusion, again, in PGEX, we have p-tag promoter. There is Shine-Dalgarno sequence, which we do not see actually at the beginning to ensure proper translation, I mean, translation of GST. And then in frame, we clone our gene of interest, either with its own stop codon, but in our PGEX, for example, we have 3 stop codons in 3 different open reading frames, so the translation should be always there, always okay. All right, other protein tags. So generally we have big protein tags and small protein tags. The most popular big protein tag, I think, is GFP, actually, so the fluorescent proteins. Then we have GST, and there is also maltose binding protein, which is also used. Then the eluting substance is maltose, and your resin is amylose, like agarose, for example. So this active compound is amylose, and you elute with maltose, just with a sugar. So it's also quite a cheap system. And in some cases, maltose binding protein, which is of a similar size to GST, it works also quite well. Okay, and then you have fluorescent proteins like GFP, but GFP is not a perfect tag as regards folding. Folding of GFP may be problematic, so still you can use it for pull-down experiments, but not to purify a protein just, for example, for crystallization or any in vitro studies, okay? So usually when you want to keep GFP and just use GFP for some purposes. GFP is also quite hydrophobic, and it may bind to the beads, agarose, for example, unspecifically, just by unspecific interactions, hydrophobic interactions. So this should be also kept in mind. And if we don't need a large protein tag, so usually when we have small proteins, we choose bigger tags. But if we have, let's say, normal size proteins, so like larger than 10 kilodaltons, we decide to use small protein tags. Like here we have polyhistidine, hexahistidine, ISO 8 histidine residues right now, historically pentahistidine. So histag is a, let's say, cheaper version for purification. It has high affinity, so it gives you high yield of your protein, but also the background is usually quite strong. So we use gradients for elution, and some proteins tend to hide the histag, so it's not accessible very well to the resin. And then it may happen that the binding is, for example, very weak, and the protein just falls out from the beads during washing. It may also happen. This has to be always checked experimentally first. And for the histidine tags, you have two types of a resin. In one, you have the metal ions like nickel or cobaltium, which is bound, which is coordinated, so it's not covalently bound. And then you have like NTA resin, or you have covalently bound ions like Ni. There is, I think, TED. So it depends on the chemical groups that are used to bind your nickel ions. And then you have like different types of resins. So polyhistidine and histag, if you want to produce high amounts of proteins, this is still, I think, and also if you need denaturing conditions. So if you expect that your protein anyhow will be insoluble, you want to denature it, and for example with guanidine salts, and you want to renature it, like gradual removal of guanidine and like renaturing conditions, then histag is good because the resin is insensitive to denaturing conditions. If not nickel, because you, for example, want to use your tagged protein for interaction studies like immunoprecipitation, or you want to monitor its trafficking inside the cells, then we choose other types of tags. For still purification, we have strep tag, which is quite popular nowadays. So it can be used only for non-denaturing conditions. It gives a lower background. So it's just another possibility. There are also antibodies, anti-strep, which recognize this strep tag, and you see the sequence of the strep tag here. Then we have other types of flags. There is MIC tag, which is not mentioned here. So a tag coming, which is originating from the oncogene CMYC, and it has a sequence. It's hanging over my desk, which is a bit longer than the other two, which are here, so HA and flag. And it's also quite polar and charged. So it's introducing charged amino acids. For some protein structures, this is not good if you want to use introduce charged residues. But still, it's quite popular. So MIG tag, I think it's even more popular than FLAG tag and HA tag still. And then we have HA, which comes from influenza hemagglutinin originally. This is the sequence. The sequence is more hydrophobic. I mean, it's mixed because epitopes generally are not hydrophobic. They have to have like polar amino acid residues. But it's good because it has those proline residues. So by the means of prolines, it can be separated a little bit structurally from your protein. And then there is FLAG, which can be single or repeated 3 times, like 3 times FLAG. And they are commercially available antibodies against FLAG or HA, which you can use for detection. There are resins, also with antibodies, so like anti-FLAG antibodies agarose, anti-HA antibodies agarose, or magnetic beads, so magnetic particles which are sometimes covered with agarose as well, but sometimes without. And they can be bound then to just magnetic, just to the magnet and fished out using a magnet. Okay, so as regards tags, I think these are the most popular ones, which I know at least. There are some modern ones which some companies offer, so really synthetic. The synthetic tags are good because the chance that such a sequence will appear in bacterial proteins or mammalian proteins is very low. So the background when we use anti-tag antibodies can be very low. So that's why it's good. The FLAG is such a synthetic peptide and synthetic tag. So usually it gives a lower background, but sometimes the antibodies which are anti-FLAG are not perfect. You really need lots of the protein. For this reason, HA is a bit better, and CIMIC is the best as regards at least detection. Okay, elution. If we have protein tags, small tags like HA, CIMIC, strep tag, FLAG, usually for elution we can use either peptide or high salt and pH. So depends on the resin. We can use that. It may be a quite tricky step, for example, if we are using pH, but usually this is for a short time. Okay, other tags, affinity tags and affinity chromatography resins that should be remembered and we sometimes use, and maybe we even don't think that this is affinity chromatography. So we have protein G agarose, which is used to purify antibodies. So when, for example, monoclonal antibodies are produced, this is a cell culture supernatant, so usually it is quite diluted. There is not that much of antibodies per volume. If you want to concentrate them, you may use protein G agarose, and then usually we elute with low pH, and then quickly neutralize, and we have like more concentrated antibody preparation. Sometimes we still use lectins. Lectins are proteins, usually of plant origin, which bind some high mannose containing proteins and other conjugates. So as regards mammalian cells, high mannose proteins are present in the ER. So they are immature proteins. And sometimes we want to fish out those ER resident proteins, and we can use concanavalin beads. There is a snowdrop lectin, for example, which is also quite specific for other types of, but usually also immature sugars, and you can use it. And there are antibody-coupled resins. There are also resins which are chemically activated, and you can conjugate your own antibody. So the antibody which you buy or you produce, and you can get a very specific resin. It's quite expensive, usually still, but let's say there are more and more reagents which enable that. Okay. Are there any questions for that part? So something else that you want to ask or share your knowledge? Because in some respects, your knowledge exceeds mine for sure.
Maybe about the start codon. Is it that in every type of plasmid we use, we don't use the start codon, or in some cases we might have to use it?
Okay, yeah. Well, it all depends on the tag. When we have, and the position of the tag. So yes, this is a very important aspect which we have to think about, is to where to place our tag. So the possibilities are that this will be N-terminus, C-terminus, or like inter, how do we say it? So we just put it inside the sequence of a protein. How to choose the position? We look at the structure if it's known, and we try to predict where the position will interfere with the protein structure the least, or we have to test experimentally. So we tag C-terminally, N-terminally, and we compare. These internal tags, these are usually rare situations. For example, when we have membrane proteins, like CD81 actually, and we want to put a tag inside the extracellular loop, for example, to detect the protein at the cell surface. And then we may try to just put this tag encoding sequence in frame inside the gene. So now, when we are using N-terminal fusions, so the tag comes first. Then the start codon is in front of the tag, and after that, we avoid using start codons because bacteria are more specific. They have this Shine-Dalgarno-based system, at least the canonical one. But mammalian cells, they are more tricky. Generally, we have the scanning mechanism of translation. So the ribosomal complexes are formed with the cap on mRNA, and then they move more or less to the first ATG. But there are also so-called Kozak consensus sequences. So it is known that the neighboring residues around ATG codon are very important. And sometimes the first ATG may not be the one that is actually used most frequently, but it can be like the second one because just the neighboring, the context sequence is much better. So it is still better to avoid the ATG codon of the gene. We remove it, okay? You can remove it when you isolate the gene by PCR. That's the most, I think, the easiest way if you do not order a synthetic gene. So synthetic gene or PCR or restriction sites, if there are suitable, good restriction sites that you can use just to remove ATG, but this will be very rare, okay? And then the stop codons come after your protein. If we have C-terminal fusions, so your protein of interest comes first, then use the original ATG of your protein. Plus, you have to think about the neighboring residues. For mammalian cells, also other eukaryotic cells, sometimes we put Kozak sequence. So there are consensus sequences for human cells, but other organisms will have their own, and we just add Kozak sequence. For humans, this consensus Kozak sequence is GCC, GCC, ACC, then there is ATG, and the Kozak consensus sequence says that if there is guanine G after ATG, it's still strong translation, even if you don't have anything in before. But canonically, we just add GCC, GCC, ACC in front of the gene. So if we tag at the C terminus, of course, we have to remove stop codons. We add the tag and then stop codon at the end. So generally, that's how it goes. Another rule when you choose the position of a tag, there is a problem with the secretory mammalian proteins, with the type 1 membrane proteins, the ones which have N-terminus outside, which are recognized by the signaling peptidase. So in order to get into the ER, eukaryotic proteins have a very specific N-terminal sequence, which is recognized and cleaved during translation when the protein just emerges in the ER. And then if we want to tag N-terminally, you have to tag after the signaling sequence. So the signaling sequence of your protein will come first, or you can use signaling sequences from some other proteins, like for example from immunoglobulins, signaling sequence are used. Then the tag, and then your protein. And you have to make sure by using predictions, so programs which recognize signaling sequences, whether when you add your tag after the signaling sequence, if it's still recognized by the signaling peptidase or not. So you cannot interfere with that. Otherwise, you will get a wrong localization of your protein. So generally, that's it. So any other questions as regards tagging? The choice of a tag usually comes also, depends on what you have in your lab. So what experience the people have, what kind of resin you have, antibodies you have. They're like favorite tags for each lab, or like even each researchers. And you just test experimentally which works good for you. Okay, so then just a short reminder about the fluorescent protein tags, which are usually used for monitoring of localization, also for the studies of protein folding and stability, because as I mentioned, GFP has some requirements as regards folding, and it will start glowing only when it's folded, because it needs its specific structure, this beta-barrel structure, which is forming a hydrophobic cage for the fluorophore, which is inside. So this can be also used. Plus, we have so-called GFP traps, so antibodies against GFP, which are bound to, for example, agarose or magnetic beads, to fish out GFP-fused proteins. Usually, GFP traps are using so-called llama bodies. So, have you heard about llama bodies? The antibodies that comes actually from alpaca and not from llama, but they are very similar. So what are nanobodies, or let's say alpaca antibodies? What are the specific features of those immunoglobulins? Anything that comes into your mind? Generally, they are smaller and they give lower background. They don't interfere, for example, with the mass spectrometry experiments. So for mass spec, those llama bodies are quite popular. So generally the camelid immunoglobulins, they are smaller and they give just smaller background. They can be more nicely separated, because people who work with immunoprecipitation, for example, or with mass spec, they know that immunoglobulins have very nasty sizes, like immunoglobulin G. These are two chains, so-called heavy chain, which is around 45 kilodaltons, and light chain, which is around more or less 20 kilodaltons. So if we have cross-reacting antibodies, they will detect immunoglobulins and they will just, interfere with our experiments sometimes. Okay, so we may use llama bodies, so alpaca anti-GFP antibodies in GFP traps, for example. And then we add fluorescent proteins as tags, either on N-terminus or C-terminus, very rarely even intra-protein. And we want them to fold, so linkers are very popular, either flexible linkers or rigid linkers, so alpha helical linkers. And then the proteins are supposed to glow and function. So both domains should be functional, the fluorescent protein and our protein. So these are the most common fluorescent proteins that we may use. Plus there are many variants. But basically there are 3 proteins coming from 3 different organisms. So on the left you have the first discovered, still the most popular one, which is of course GFP. So it comes from this nice jellyfish, which is called Aquaria victoria. Sorry. This is. Oh, Jesus. There is this one phone which is, I don't know how to silence. Okay, so Aquaria victoria, the jellyfish. Actually, it's not green, it's bluish because it has this fluorescent system. So there is aquarine, which is the first fluorescent protein, which is absorbing, I think, the energy and it's transferring to GFP or the other way around. But generally, GFP is just one of the fluorescent proteins of this jellyfish. And usually it's used in form of variants. So after mutations like enhanced GFP, with more brighter, more stable fluorophore. There are also mutations which make GFP monomeric because those fluorescent proteins have a tendency to dimerize. So when you are choosing your fluorescent proteins, please make sure that this is like new generation, which is monomeric if you want to study protein interactions. Otherwise, you will have a bias, like a background just from GFP itself. But I think the most modern variants, they have this M in front of the name, and it stands for monomeric. Okay, so the 3 proteins are they have very similar structure, as you may see. So the second one is a red fluorescent protein coming from this anemone, so this sea coral, which is called Discosoma. It's Discosoma species because there are different species of Discosoma which can be used for isolation of the original gene. But we are using mutated versions of RFP usually, and M-cherry is the most popular. It gave the origin to so-called M-fruit proteins, so with different fluorescent emission spectra, so different colors, and you can choose the one that you like. As regards the structure, M-cherry has a more tricky structure. It's a bit more stable, but it has more problems with folding. So when we are using M-cherry fusion, sometimes it affects the structure of our protein and the function of our protein. But for example, it is not sensitive to lower pH and oxidation like GFP is. For example, when people want to track proteins in lysosomes and also autophagosomes, there is a problem with pH. GFP is sensitive to this pH, it stops glowing, and M-cherry is still active. So they use M-cherry for fusions. And the third protein, which is at the bottom, I think it's overtaking the market. It's called neon green, and it's a variant of a green fluorescent protein coming from the sea lancelet, Branchiostoma lanceolatum. It's also green, it's brighter than GFP and more stable than GFP. It had to become monomeric, so it's also changed by mutations to be monomeric. And I think all the other names are just variants of those three. Like, for example, we have clover, which is a variant of GFP, and we have M fruits, which are derived from red fluorescent protein, monomeric red fluorescent protein, like we have D tomato, we have M orange, we have M plum, far red, M banana, and so on. And we have blue versions of GFP, which are also used. There is another, of course, application for such fusion proteins, which is FRET. So this energy transfer experiments, when you study protein interactions, so you have two fluorescently tagged proteins, but the set of fluorescent proteins has to be very specific. So you induce one of them and the other one emits light. There is also an application which is called FRAP, so frequency of recovery after photobleaching, when we are using GFP and we want to study, for example, movements of a protein. Because we bleach GFP and we measure how fast the signal is recovered. So new proteins, fluorescent proteins, for example, move to this compartment of a cell. So generally that's it. Okay. So I think this covers the protein tags. And now some hints how to remove small substances like glutathione from our samples. Or for example, imidazole. When you are using nickel, so histag and nickel or cobaltium resins, and you usually we elute with imidazole, which is resembling the histidine rings, and it's just used for elution. Then we want to get rid of imidazole, and how to do it? Well, we of course use the knowledge about the size difference usually. So these are some most common methods. I think the most historical is size exclusion chromatography, or SEC, or gel filtration, when we are using porous resins, which can be actually agarose itself. So cepharols can be also used, maybe not for proteins because it has quite big pores. But Cefadex, so dextran, has smaller pores, and you can just choose different pore size of the resin to separate proteins with various ranges of size, like smaller proteins, bigger proteins. So this is the principle, just to remind you. Smaller proteins usually just intercalate, they just get into the beads, they interact with the beads, and they are retained on the resin, on the column. And the bigger particles just move more quickly, they move first. And we can buy also desalting columns, which are based on SEC. They are called PD10 usually. So these are PD10 columns, which can be used for desalting, for example, if we want to remove salts or just change the buffer that our protein is in. Another popular method is still dialysis. So we have to put our sample in a bag. Actually, this is the cellulose-based polymer. It's cellophane in Polish. I don't know what's the name in English of that, but it must be quite similar. So it's porous. And we may use those foils, those membranes with different size of the pores. And this parameter is called MWCO, so the cutoff, molecular weight cutoff. And they are membranes, dialysis membranes with a cutoff of, for example, 3 kilodaltons, or even smaller, when the proteins just stay inside this tube. And because of the gradient, I mean this concentration difference, the hypotonic buffer is usually around, and then those smaller molecules, they just get out of the membrane and the proteins just stay inside. So this can be used also for glutathione removal. And we prepare those dialysis tubes ourselves sometimes because it's cheaper. So we just buy tubes. Usually they are sold as long tubes, which we cut with the scissors for the desired length. And we have to use clips to close it at the beginning and at the end. And then we just put it into a big beaker with the buffer, and we rotate overnight. We change the buffer, and at the end, we will have our protein inside this new buffer, for example, without glutathione. You may also buy dialysis cassettes when they're like plastic frames and the membrane is already inside. But usually this is for small volumes, like if you want for antibodies purification, people use it. But for bigger volumes, usually this is just not sufficient. There are also new methods of removing small molecules which are more commercial and for bigger volumes. But for the lab work, usually we use that. And I think the most popular now, just because they are the easiest way, this is the so-called ultrafiltration. So when people talk about ultrafiltration, usually they think about those centrifuge tubes with the membrane, like Amicon filters. Amicon, this was the original name, trade name from the Millipore company. Now Millipore, I think, is not Millipore anymore, but and you can buy those tubes in different companies. They may have different names, but the principle is very similar. So you choose either small tubes, small centrifuge tubes, or bigger ones. So this is like a big Falcon tube, but they are also even larger tubes. But then you need a specific centrifuge with the buckets that would fit those bigger tubes. So this is the only problem. But for the standard ones, we have either those 15 milliliter tubes or 30 milliliter tubes, or this is for 4 milliliter volume, which you can use for concentration and desalting, because you may either concentrate your sample in the same buffer, or you may remove the whole buffer. Usually there is a very small volume that stays on the membrane, and then you may add, for example, only PBS buffer, centrifuge again, you may repeat this step. We lose some of the protein, even though there is still this molecular weight cutoff, so specific pores of the membrane, like, for example, 3 kilodaltons or 50 kilodaltons. You want to remove small proteins. You want to just leave big proteins inside. But still, there are some materials which are leaking, so like small proteins, but some bigger proteins still going through the pores. Sometimes they stick to the membrane as well, so may lose some of the material, but usually it just really works to concentrate and dissolve your material. Okay, anything else? No, this is I think the end. So I'm just thinking if there is anything that should be covered. I was talking with my group about antibodies. So I don't know if all of you know what's the source of antibodies in the lab that we use. For example, anti-CD81 and anti-GSD antibodies that you are using, how are they produced?
By immunizing animals?
By immunizing animals, always, almost always, because we may buy now recombinant antibodies, which are produced in cell cultures, in mammalian cell cultures, and They are obtained by genetic methods, but then still you have to know what's your epitope. And usually we know what's the epitope, I think still originally using the phage display technique. So we just learn what's the sequence of the epitope, or by sequencing, this epitope sequencing, we know what's the sequence of the epitope. And then we can clone this epitope encoding fragment into specific plasmids. We transfect specific mammalian cells which are used for production, and then the usually single chain recombinant antibodies are produced this way. And then, but this is still quite rare as regards just standard applications. So by immunization of animals, but your antibodies actually were prepared, let's say, in more sophisticated way. So after immunization of these were mouse antibodies. So after immunization of mice, their spleens were removed, and so-called splenocytes were isolated, which are B cells, among all, but mainly B cells, and they were fused to the oncogenic mouse cells, which are called myeloma. And these fusion proteins are called hybridoma. And hybridoma are producing antibodies. So they may be mouse hybridomas, usually, sometimes rat hybridomas as well. And you can grow them in your lab. You can buy hybridomas. They grow in suspension, and they just produce antibodies to the cell culture medium. And you collect the cell culture medium, and you can either purify your antibodies using epitopes, or you do clonal selection. This is actually during production of hybridoma. So they are monoclonal because they were separated into single cells. Each cell was giving rise to a clone, so the culture of a cell, and each produces one type of antibodies. They're called monoclonal antibodies. Another option is just immunization of a large animal and just collecting their blood and their serum fraction. And usually then directly serum is used to detect antibodies, and they are polyclonal antibodies usually. If you immunize a rabbit with a peptide, then you can get monoclonal antibodies, rabbit monoclonal antibodies, but usually they will be polyclonal. Bigger animals, because this is more economical, rabbits, but more often big animals like donkey, goat, alpaca. So lots of blood, and without killing the animal, so just gradual collection. Okay, so this is how secondary antibodies are produced, by immunization of a larger animal with immunoglobulins. Then it's either a serum fraction, but usually these are purified antibodies, like IgG fraction, which is obtained. Okay. Are there any questions, generally, that you want to ask about the test? About the test, so we agreed with other supervisors from the groups that you will write the test together on big aula, so in the big lecture hall. And it's up to you when you want to do it. So you have to choose one date, one time when we will book this lecture hall. And all the supervisors, we will prepare the questions for you together. And they are usually based a little bit on the theoretical knowledge or explanation of the procedures, what happens during the lab. Sometimes small troubleshooting, like I'm performing Western blotting, I have no bands. What could go wrong? Or I'm performing Western blotting and I have many bands. What's the reason for that? Or we are purifying lysozyme and we have no lysozyme in our fractions. What's the reason? What's the potential reason? Okay, so there are usually several explanations. These will be open, short, open questions. This is the original way that this test was written, and we are getting back to it. So short, open questions. And we will prepare the test in two versions, either English or Polish, and you can choose which one you want to answer. Okay? One question for sure will be about the safety rules. And this one question you have to answer correctly. You have to pass it because we have to evaluate your knowledge on the safety rules. Okay? Usually it's not that difficult, but just please read the instructions once again. I have a rule that usually I want all the reports to be returned to me before the final test. Just returned. So until the midnight of the previous day, I just want to get them on my email. So, but I have to still send the data to my group. So it's there is still some time. Okay. Are there any other questions? So think. If not, you can always write me an email. You can try on Teams, but I'm just checking my Teams more rarely now. We don't have that many lectures and seminars online anymore, so you can write me just an email with a question. Okay, if not, then thank you very much for your attention and good luck during your classes. And there is still one piece of knowledge which we do not cover. This is the ELISA. But ELISA is very well explained in the instructions. Like there are 3 types of ELISA tests which we are using. You can just try to think about potential applications. So where do we use ELISA test? For diagnostics, for example, or food industry. More and more often we are using small Western blotting, let's say, devices for diagnostics, like, for example, COVID antigenic tests. They're based actually on Western blotting, or kind of Western blotting, because the protein, antigenic protein, or antibodies, depending on what you want to detect, are placed on a membrane, and by just capillary movement of a solution from sputum, serum, sometimes from blood, or sometimes urine, you just give a source of antibodies or antigen for detection. And the material which is immobilized on the membrane is also coupled to antibody, coupled to to the chromophore. You have like colored reaction. Okay, no more questions? Then thank you very much and see you in the classes.
Goodbye.
Thank you very much.
Bye-bye.
Goodbye.
Thank you.
Thank you. Goodbye.
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 Bye.